Pick any population on Earth that's been scattered across 4 continents for 2,000 years, and population genetics has a rule for what should happen to them. They should blend in, not slowly disappear exactly, but dissolve, generation by generation, into whatever populations happen to surround them, until the original genetic signature is buried so deep under centuries of intermarriage that no test could ever pull it back out again. That's the rule. It's held up case after case, population after population, all across the world. Then geneticists tested it against one specific population, and the rule broke. 1,000 years of living among Europeans left a smaller genetic mark than 3,000 years of shared ancestry in a place most of those same people had never once set foot in. That's not a loose interpretation of the data. That's what multiple independent research teams using different methods and different sample sets kept finding over and over, no matter how they ran the numbers. Stick around, because by the end of this video you'll understand exactly how a population can hold on to its genetic identity for 2,000 years without ever having a shared homeland to anchor it, and why the answer traces back to something as small as 350 people huddled somewhere in medieval Europe. If untangling stories this precise, this old, and this genuinely strange is the kind of thing you want more of, hit subscribe right now, because that's exactly what this channel does every single week. Let's start with the basic expectation, because it's what makes the actual result so strange. When researchers began serious genome-wide comparisons of Jewish populations in the early 2000s, the working assumption was straightforward. Ashkenazi Jews, whose ancestors lived across Central and Eastern Europe for roughly 1,000 years, should look genetically similar to the Germans, Poles, and Russians they lived alongside for that entire stretch. Sephardi Jews should carry a strong Iberian and North African signature. Communities settled in Iraq and Iran for 2,000 years should carry a heavy, regional Middle Eastern signature layered thick on top of whatever came before. That's simply how population genetics is supposed to work. Geography, time, and intermarriage blend populations together. They smooth the edges. Eventually, they erase the origin point almost entirely. That's not what the data showed. Autosomal DNA studies, examining the 22 non-sex chromosomes that carry the bulk of human genetic information, consistently show high genetic relatedness among Ashkenazi, Sephardi, and Mizrahi Jewish populations corresponding to a shared Middle Eastern ancestral core, with regional admixture layered on top of it rather than replacing it. Communities separated by oceans, by different everyday languages, by centuries of independently evolved food traditions and religious customs, still point back to the same underlying origin once you strip away everything accumulated on the surface. That origin traces specifically to the Levant, the strip of Mediterranean coastline covering modern Israel, Palestine, Lebanon, Syria, and Jordan, sitting directly at the crossroads of Africa, Asia, and Europe. Multiple studies have found that Jewish communities worldwide share more genetic commonality with each other and with other Levantine populations, including Druze and Palestinian samples, than with whichever population they spent the last 1,000 years actually living beside. Sit with that for a second, because it genuinely inverts the normal expectation. 1,000 years living among Europeans left a smaller genetic imprint than 3,000 years of shared ancestry in a homeland most of those same people had never personally visited. That's not how assimilation is supposed to behave. But the story gets stranger once researchers started drilling down into the specific branches of the diaspora, and the Ashkenazi branch in particular turned up something close to a genuine mystery. Modern models of Ashkenazi ancestry describe a population shaped by a specific, severe genetic bottleneck sometime in the medieval period, a moment when the entire founding population contracted down to somewhere between 330 and 350 individuals. Not a typo. 350 people, smaller than the student body of a single school. Research published in Nature Communications found that population was, at that founding moment, genetically close to an even mixture of Middle Eastern and European ancestry, and every Ashkenazi Jewish person alive today descends from that same tiny founding group, closely enough that researchers describe the entire modern population as related at the level of 30th cousins or closer. Not a figure of speech. An actual, measurable genetic relationship detectable directly in shared DNA segments passed down from that same small founding population, estimated to have gone through this bottleneck somewhere around 600 to 800 years ago, followed by explosive population growth in the centuries that followed. Here's what happens when a population goes through a bottleneck that's severe and then rapidly expands back out from it. Random genetic quirks carried by those original founders, quirks that would normally average out and disappear in a larger, more genetically diverse population, don't get the chance to dilute away. They get carried forward, amplified across every subsequent generation instead. Geneticists call this the founder effect, and in the Ashkenazi population specifically, it produced something that has fascinated and troubled researchers studying it for decades: a measurably elevated frequency of specific hereditary conditions. The numbers are precise, and they're still clinically relevant today, not simply historical trivia. Roughly 1 in 27 Ashkenazi Jews carries a Tay-Sachs mutation, compared to roughly 1 in 250 in the general population. Around 1 in 10 carries a Gaucher disease mutation, compared to roughly 1 in 100 elsewhere. Altogether, the founder effect elevated the frequency of somewhere around 16 distinct inherited conditions within the Ashkenazi population specifically, several of them severe neurological disorders. Here's where the science takes a genuinely unexpected turn, though. Several of these conditions, including Tay-Sachs and Gaucher disease, share something in common at the molecular level. They all involve defects in how the body stores and processes a specific class of lipids. That clustering caught researchers' attention because it doesn't look like random chance. Some researchers have proposed that carrying a single copy of these mutations, not the double dose that causes the disease itself, but just one copy, may have offered some meaningful protection against tuberculosis, a disease that devastated crowded medieval European cities for centuries. If that hypothesis holds, it means natural selection was quietly preserving these mutations generation after generation, trading a small, tragic cost in children who inherited 2 copies for a broader survival advantage across the wider population, facing a genuine epidemic threat. Biology in this reading wasn't optimizing for anyone's comfort. It was optimizing for raw survival against whatever was actually killing people at the time. There's a second, entirely separate genetic signature layered into the same population, and it carries real consequences in hospitals today, rather than sitting purely in the historical record. Roughly 1 in 40 people of Ashkenazi Jewish descent carries a BRCA1 or BRCA2 mutation, compared to roughly 1 in 250 in the general population. Mutations tied to significantly elevated risk of breast, ovarian, and several other cancers. This isn't abstract population history. National health services in multiple countries now run targeted genetic screening programs specifically for people with Ashkenazi Jewish ancestry, precisely because that same medieval founder effect is still directly shaping cancer risk for millions of living descendants 800 years removed from the original bottleneck that produced it. Now here's where the evidence moves from inference to something closer to direct proof. A research team working with Harvard Medical School took an approach nobody had fully attempted before on this specific question. Rather than working exclusively from the genomes of living descendants and reasoning backward, they extracted ancient DNA directly from physical remains, teeth recovered from a medieval Jewish cemetery in Erfurt, Germany, reading the genetic record straight from the source rather than reconstructing it secondhand. It stands as the largest study of ancient DNA from Jewish individuals conducted to date. And the results, published in the journal Cell, revealed something researchers hadn't anticipated going in. The remains split into two genetically distinct subgroups, buried in the same cemetery, presumably attending the same synagogue, speaking the same everyday language, yet carrying measurably different genetic profiles. One subgroup showed a stronger Middle Eastern genetic signature, the other leaned more heavily towards Central and Eastern European ancestry. Two closely related but genetically distinguishable communities, living side by side in the same medieval German city that would go on to merge into what became the singular, modern Ashkenazi population, absorbing little detectable outside genetic influence across the roughly 6 centuries that followed their burial. The ancient DNA offered something else too, direct physical confirmation of the founder effect itself. Several of the same disease-linked mutations still elevated in Ashkenazi populations today were already present in these medieval remains, meaning researchers weren't just inferring an ancient bottleneck from modern statistical patterns. They were looking directly at teeth belonging to people who actually lived through it, carrying the same mutations forward in their own bones. Mitochondrial DNA analysis added another striking detail. Roughly 1/3 of the individuals buried in that medieval cemetery traced their maternal line back to a single ancestral woman. One woman, 1/3 of an entire community's maternal lineage, converging on one specific ancestor. There's one more thread in this story that seems to belong somewhere else entirely until you actually look at where it leads. A study examining mitochondrial DNA variation across tens of thousands of individuals, comparing East Asian populations against Jewish communities, found that a small number of Ashkenazi individuals carry mitochondrial lineages, properly classified as Eastern Eurasian in origin, estimated to have diverged from the broader Ashkenazi maternal line somewhere around 1,000 to 2,000 years ago. That timing lines up strikingly well with the era of the Silk Road, the sprawling network of trade routes connecting the Mediterranean world to China across more than 1,000 years of continuous exchange. The Silk Road, it turns out, carried more than silk, spices, and religious ideas across that enormous distance. It carried people too, and at least a handful of those people left a genetic trace inside the Ashkenazi maternal line that researchers can still detect and roughly date, well over 1,000 years later. None of this happened by accident, and researchers have a fairly clear picture of the actual mechanism holding it all together across 2,000 years of dispersal. The first factor is endogamy, sustained marriage largely within the community rather than with surrounding populations. Jewish religious law and long-standing communal practice have historically discouraged marriage outside the group, and the genetic data reflects that pattern directly, showing up as unusually low genetic drift relative to how much time had actually passed since the original Levantine dispersal began. The second factor is considerably darker, and it's worth stating plainly rather than glossing over. Historical persecution, ironically, reinforced that same endogamy from the outside. When surrounding societies made intermarriage with the Jewish community illegal, socially catastrophic, or simply dangerous across multiple countries and multiple centuries, those same restrictive policies inadvertently helped preserve a genetically distinct lineage, without that ever being anyone's stated intention. The specific historical forces aimed at isolating and often harming this population also, as an unintended biological side effect, helped conserve exactly the genetic continuity researchers can still trace directly back to a Bronze Age Levantine population today. Then there's the sheer scale of demographic growth layered on top of all of it. The Ashkenazi population grew from roughly 50,000 people at the start of the 15th century to somewhere around 5 million people by the start of the 19th century, 50,000 to 5 million in 4 centuries. That's not ordinary demographic growth. That's an explosive expansion launched from a catastrophically small founding base, which means every genetic quirk carried by those original few hundred founders got carried forward and amplified across millions of descendants far faster than it would have inside a larger, slower-growing, more genetically diverse population. There's a genuinely strange coda to all of this too, one that keeps surfacing in modern clinical genetics. A meaningful fraction of people carrying detectable Ashkenazi Jewish genetic ancestry today have no idea that ancestry is even there. In one dataset tracking nearly 2,000 individuals who tested positive for a BRCA mutation and voluntarily reported their own ancestry, roughly 21% did not report any Jewish ancestry at all, despite more than half of that same subgroup carrying clearly detectable Ashkenazi Jewish genetic markers underneath. The genetic signal, in other words, persists intact, even after generations of conversion, assimilation, or simply lost family history have erased any conscious awareness of where it actually came from. The biology remembers details the family history sometimes doesn't. It's worth being direct about what this body of research does not show, because the topic has attracted plenty of bad faith interpretation from multiple political directions over the years. It doesn't demonstrate any kind of genetic hierarchy, superiority, or purity in any meaningful biological sense. What it demonstrates instead is something considerably more specific and more interesting. A population shaped by an unusually well-documented, unusually traceable combination of catastrophe and continuity, where devastating historical bottlenecks, real persecution, and a genuinely unusual demographic recovery all left a measurable, still legible fingerprint directly inside the genome, readable today by researchers working with tools that didn't exist even 20 years ago. What all of it ultimately comes back to is something genuinely remarkable once you strip away every layer added on top of it since. Roughly 2,500 years ago, the Babylonian conquest of ancient Judea forced a population apart, sending one stream east into what's now Iraq and Iran, and eventually another stream west, deeper into the Mediterranean world and, later, Europe. 2.5 millennia later, the genomes of their descendants, scattered now across dozens of countries on nearly every continent, still carry a measurable trace of exactly when and how that original separation happened, preserved not in any single document or oral tradition, but directly in shared segments of DNA that a laboratory can still read today. If untangling stories this precise, hiding directly inside a population's own biology, is the kind of thing you want more of, subscribe to DNA Exposed, because next week we go looking for another population whose genome remembers considerably more than its own written history ever managed to record. We'll see you in the next one.